Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Christopher Sucato and Cynthia Swanson from Charles River Laboratories.
Thank you, Leah. It's a pleasure to be able to present this webinar today on AUC and gene therapy, with a focus in particular in our Charles River Laboratories, our site in Shrewsbury, Massachusetts, where I'm coming to you from, with a focus on AUC and AAV platforms. While in the past, several mechanisms for gene delivery have been advanced as potential therapies and have held the promise of a stable and long-term incorporation of vector DNA into host cells, it's safe to say that at present, adeno-associated viral vectors, or AAVs that is, are indeed the leading choice for gene therapy treatment of a variety of conditions. And AUC, meanwhile, and the size distribution profiles that derive from the sedimentation data are becoming more and more a key measurement in the AAV development process. What's contributed recently to the success of AAV-based strategies are some advances on the discovery and R&D side. One, in developing recombinant AAVs with clinically advantageous properties, optimized transgenes that are efficiently delivered to the host cell nucleus and are reliably translated. And two, on the process and manufacturing side, likewise, advances there that have made AAV a practical drug material from a commercial perspective. Now, these developments scientifically are of great value and interest, I'm sure, to this audience and to the field in general, while a deep dive into many of those aspects would be beyond the scope of this webinar. I'll present a collection of references. We'll show a collection of references at the end here for anyone who may be interested. Nevertheless, some of the recent advances in AAV technology can indeed be understood in terms of the schematic shown here, illustrating how good manufacturing can lead to a product with an improved profile in terms of the empty and full and other species, shown at the top part of the diagram here. A critical quality attribute, incidentally, which will be a focal point for this presentation, of course, but also other favorable capsid properties, including the VP1, 2, and 3 protein makeup gained by recombinant expression optimization, which can guide tissue specificity of initial receptor-mediated endocytosis, shown here in the middle part of the diagram, and the endocytotic process in the cytosol overall, and likewise favorable properties of the transgene, allowing for packaging the maximal gene size and efficient, stable incorporation in the nucleus to the bottom part of the diagram. The timeline shown here makes a good case for why discussions like these are particularly well-timed. It is only recently, after all, that clinical applications of AAV have begun to take off commercially, development pipelines expanding in earnest really only in the last decade, and only 2 FDA-approved products at present, but with many, many more in development pipelines globally. And regulatory guidance on these CQAs that define a safe and effective AAV product still taking form in many respects. In this talk, we'll discuss, and importantly also show some data, in particular, how recent advances in AUC characterization of AAV products may help inform that type of guidance. Now, in the past, at presentations like this, I've sometimes gone into some depth about the history of AAV, its wild-type traits, etc., that have led up to the present-day status of AAV as a gene delivery platform. In the interest of time, I'll shorten that part somewhat here, but suffice it to say, there are indeed plenty of review sources for this kind of historical background. Nevertheless, let me say briefly that wild-type AAV, once thought to be simply a contaminant of adenovirus preparations, illustrated here in this micrograph, It was first characterized extensively as early as the 1960s as a unique viral species, these AAV particles. It was shown to be replication defective, going through its full replication cycle only if the host was co-infected with a suitable helper virus, such as adenovirus or herpes virus. An important observation at the time, though, was the finding that AAV itself only caused a mild immune response, such as the generation of neutralizing antibodies, and infection did not produce any observable disease condition in these wild-type AAVs. So the apparent lack of pathogenicity in the wild-type was a main advantage that would be leveraged in the development of recombinant AAV in gene therapy. Other advantages are related to the infection cycle in the wild-type. For example, in the latent phase, AAV integrates itself stably into the genome at only a single specific chromosomal site. And secondly, AAV has a good infectivity rate with quiescent or non-dividing cells. So these advantages are in contrast to other platforms like retroviruses, which may present a random insertion and mutagenicity risk, and are generally less effective in these non-dividing cells. Now, structurally, the wild-type AAV virion itself is a small, non-enveloped icosahedral particle, only really about 20 nanometers in diameter. And so, incidentally, one of the few disadvantages is due to this small capsid geometry and the relative size of the gene that can therefore be packaged. The AAV capsids in the wild type, contain a linear single-stranded genome of about 4.8 kilobase pairs. And wild-type AAVs are replication defective, as mentioned. In other words, they are dependent in the wild type on co-infection with a helper virus such as adenovirus in order to complete the AAV replication cycle. Yet, recombinant AAV has several advantages as a gene delivery vector. It is safe, it is not known to be associated with any disease symptoms. It's effective, showing long-term transgene expression, and it's robust. It's being known to be, in many cases, heat stable and showing stability under storage conditions, etc. Now, although the presence of a helper virus is a requirement for AAVs in the wild type, when developed for use as a gene therapy vector, production of recombinant AAV can be accomplished by the so-called helper-free methods, where the necessary helper factors are instead co-transfected into the production cell line. Additionally, in most production schemes, like this quite well-known and common triple co-transfection schematic shown here, the wild-type AAV genes for replication and for the structural capsid proteins are also co-transfected, and the rep and cap genes are removed from the viral target DNA, and only the therapeutic transgene sits between the inverted terminal repeats. So there are a few different ways to accomplish this endpoint, which I show here at the bottom right-hand side of the diagram. In other words, the purified drug substance as an endpoint, as it were. And as it turns out, the exact modes of production and purification employed in the manufacture of recombinant AAV can have an important influence on attributes that appear to be critical to the quality of the final product. One such critical attribute is related to the nucleic acid that is packaged within the capsids of recombinant AAV. When produced from transfected cell lines, it has been shown that recombinantly produced AAVs are not uniform with respect to the packaging of DNA into the core. A higher percentage of capsids may contain no genetic material at all and would be termed empty capsids, as I show here. Additionally, capsids have been shown to contain incomplete portions or fragments of the transgene coding DNA that target DNA, or even non-target extraneous nucleic acid contaminants. So these observed empty or partially filled variants of recombinant AAVs can be dependent on the production and purification method and are a critical quality attribute because recent reports suggest that the extent of empty or partial capsids in an AAV drug product may correlate with vector-related side effects and reduced gene transduction rates physiologically. Yet other reports hint at possible benefits derived from having at least some percentage of the capsids as empties. which is an interesting potential observation. And so, as I mentioned in the very first slides, a big part of gene delivery characterization at Charles River is done nowadays by analytical ultracentrifugation, and I look forward in just a few moments to getting into some of the data and the discussion associated with that. But I feel I must spend a little time here on the following question, which is shown here on the left, because it is something that comes up all the time in our interactions. The question of just how to treat these capsid-related impurities that invariably show up in AAV drug products, empty capsids being the most abundant in many cases, but other variants as well. Essentially, how should the acceptability limit thresholds for these species be set? What's clear from guidance documents is that levels of empty capsids and other capsid-related variants should be closely monitored. That much is clear. Unfortunately, there's no one answer as to what levels of these impurities or contaminants would be considered an acceptability threshold in specific drug products. And these product-specific assessments of the empty-full ratio, etc., as well as the presence of partial species, should be done in correlation with toxicology and potency on those specific products in order to justify any acceptability limits on the empty-full threshold that one might get from an assay like AUC. Now, while our focus in the biophysical group here at Charles River is the validity of the assay itself, the AUC assay, in other words, these assay parameters, are they precise, linear, accurate, etc. We're focused on that, but we understand the importance of doing this work with some sense of where it all fits into the overall regulatory approval picture. And so I've included here a few references towards that end. This reference will certainly be of value to anyone interested in regulatory guidance. This FDA publication from January of this year is the latest guidance on CMC best practices, and section 32P223, which I outline here and references therein, gives the reader a sense of how empty levels should be approached, how the regulatory agency is beginning to approach these. Just how to think of empties, though, is still an active area of development, and this current opinion piece, which I show here, still quite contemporary, presents some case for making the best of drug products that do contain significant levels of empties. Whilst here, in a review from Wright at UPenn, some of the downsides that empties may present to AAV therapies and the case for keeping empties low is covered. Lastly, I have a link here to a very informative discussion that was part of a virtual think tank series recently, and has some thoughtful insights into how regulatory thinking may be evolving on the topic of gene therapy overall. A good reference to consider. And certainly, as regulatory thinking is evolving, perhaps not just evolution but revolution is at hand, judging by the development pipeline for AAVs. While, as mentioned, only 2 AAV therapies are approved at present, a great number of candidates will enter into clinical trials in the near future, representing a variety of AAV serotypes and targeting a wide range of disease conditions. Certainly, given the rationale that the species distribution profile of these diverse products should be assessed carefully in accord with toxicology and potency data, the presence of empties, etc., should be considered individually for different drug products. The importance of methods like AUC becomes quite apparent, which I think makes it a good time to show you just indeed what AUC can do. Now, the AUC assay itself is by no means trivial in terms of its setup, execution, data analysis, and we could spend an entire webinar just on the fundamentals of the sedimentation process itself, the instrument parameters and the data deconvolution, etc. Suffice it to say, the new optimal model of AUC, recently put out by Beckman Coulter, presents the opportunity for improved analysis, faster scanning, easy setup of dual absorbance and interference detection, and potentially improved signal-to-noise and resolution as well. That's the new Optima imaged on the left. Rotor and cell parts, towards the middle of the diagram here remain largely unchanged at present from their original inception. And the fundamental data is also shown here, collected in an AUC experiment. These are the sedimentation profiles shown here, generated by spinning these special cells at a high centrifugal velocity, causing sedimentation of heavy species like viral particles. If a sample is highly monodisperse, a single boundary region or inflection region will appear, and the travel of this boundary from the top of the cell to the bottom over time is a function of the rotor speed and the mass of the species that's being sedimented. If a sample is heterogeneous, however, each of the sedimenting species in that heterogeneous mixture may contribute individually on its own, separately to this raw data profile, in the form of a superposition of several different boundaries, each boundary proportional to the abundance of the individual species and the mass of that species being sedimented. Numerical fitting algorithms take this raw sedimentation data and deconvolve it into a peak profile, a peak distribution profile, which we can readily observe and quantify. Now, I've gone through these basics rather quickly, I admit, in favor of focusing the remainder of the presentation, or my part of it, showing some real results. Many resources, however, are available for a deeper dive into the fundamentals of this assay. A great review is one from Professor Jim Cole at the University of Connecticut, which I do reference here. But here, folks, on this slide is what real AUC experimental data setup looks like. I should note that these data shown here, and all results I'll show hereafter, derive from R&D AUC samples. These are not data from clinical samples or drug products, but are instead simple R&D AAV samples that nevertheless are a good representation of AAV behavior. For this sample, which I will refer to here simply as sample A, we loaded experimentally, in terms of setup details, we loaded about 150 microliters at a titer of 1e to the 13th viral particles per mil. That was the loading volume. The total experimental volume in the cell was closer to half a milliliter. Centrifugation was performed at 20,000 RPMs, and detection was at 230 nanometers UV absorbance detection. As you can see, the sample concentration in the instrument was about 0.5 ODs. You can read that carefully off of the y-axis there. And we collected about 60 scans, which collected over the course of about 4 hours. The results here are a series of sedimentation profiles collected over time, each trace representing essentially a single time point, and the data density can be thought of as how closely spaced these traces are. The movement of these boundary regions gives you some sense of the sample heterogeneity and the relative size of the species that are being sedimented. In the case of this sample, because of the multiple boundaries, clearly this is not a monodispersed sample. You can sort of see that there's a waviness and not just a single boundary happening here. So indeed, there is potentially more than one species present. Contrast the results on sample A with those of sample B, which I show here. Again, this was a setup very similar to that of sample A. The sample was run on the AUC almost exactly the same way, and we have the raw data set shown here. Yet some subtle differences in the raw data appear even by eye, comparing sample A and sample B. When the fitted results are observed, as shown here for sample A and here for sample B, the difference between the two is more readily interpreted, and the value of AUC becomes clear. Observed here at 80S on the x-axis, a peak which is intermediate between the empty and the full capsid species. This may correspond to a partially filled capsid species, or perhaps some other type of variant, but nevertheless, what's clear from these two AUC assays is that the method is capable of resolving and quantifying the relative abundance not just of empty and full, but even intermediates with excellent baseline resolution, and can discern the difference between relatively small percent abundances. And so this is a critical advantage in AUC in being able to make these kind of high-resolution measurements and discern subtle differences. If you take anything away from this presentation, it would be the difference between these two slides here in terms of what AUC can do. Now, these may be two different serotypes as they are in this case, but if you are developing an AAV product and are comparing two or more samples as a result of a process change on a drug product, the difference between a result like this and this may be very significant to your development decisions. While some other methods like capillary electrophoresis and ion exchange HPLC are being developed for empty full resolution, at present there is no method that we're aware of that can surpass the kind of resolution demonstrated here for an AAV sample, showing excellent baseline resolution not only for the empty and full peaks, but as I mentioned, intermediate species as well. Now, the ability to resolve subtle differences in the abundance of small peaks, as I've just shown, would only be reliable if the method had sufficient precision. And of course, precision is a primary objective for any method validation. So we've investigated what, in our experiments here at Charles River, we've investigated what is the expected precision obtained for AUC assays on AAV. I show here a set of n=6 runs done by the same analyst on the same day on an AAV sample, and the statistics for the sedimentation coefficient and the peak area indicated here indicates that precision by AUC is fit for purpose, having RSDs for both sedimentation coefficient and peak area that are quite respectable. Now, admittedly, chromatographic methods are able to obtain higher precision levels for some of these peak characteristics or the analogous peaks in HPLC. Higher precision levels than AUC by HPLC, but the kind of repeatability shown here in the numbers and the relatively good overlap between replicates, evidenced visually by the graph that I show, would tend to argue that AUC is indeed very suitable for quantifying empty and full levels, especially given the very good peak separation here, the baseline separation between peaks, which is difficult to obtain in the case of AAVs by certain chromatography methods. And longer-term variability shown here as well, as we've done by intermediate precision attempts, whereby the same assay was run on a second day, and again supports the notion that the AUC assay, when applied to AAV samples, will have the kind of precision that you can count on for a validated method. Another parameter we've evaluated in our quest to determine if AUC can meet validation standards is linearity, which I'll cover here. We've actually evaluated the linear response of AUC in two different ways. First is by the means I show in this slide, as I illustrate here schematically on the left, where we have run AUC on a series of samples. We've created this series by starting with the so-called full capsid stock, the drug product species, if you will, as a baseline, and then we titrated in known amounts of a different sample stock, which is enriched in empty capsids, essentially a pure empty capsid sample. By adding known amounts of empty to a fixed and also known amount of full capsids, we have the ability to challenge the AUC assay in its ability to have a linear response to a change in the amount of one species in the distribution relative to another, which is remaining relatively fixed. The data here indicate that indeed a good linear response is achieved, and this is a significant accomplishment, I must say, for AUC, a significant accomplishment, because this result, together with a reasonable value for precision, gives the analyst confidence that small differences in the empty and full ratio will indeed be picked up by this instrument, by this AUC assay, which is what you want to know. If your process change is having a change in empties, etc., you will see it. A variation of the linearity challenge to AUC is the ability to show a correlation between the total integrated area under the curve as a function of the overall amount of sample loaded into the cell, which we show here. We've evaluated this kind of situation by taking a stock amount of sample and making a serial two-fold dilution series. When we run that sample set in an AUC, again, we see a nice linear response between the overall concentration loaded into the cell and the total integrated area under the distribution in area units. This is good because it shows the concept of recovery in an HPLC sense, this concept of recovery holds true for AUC in a similar way to how it does for chromatography. Now, knowing the limits of detection in an AUC assay will also be important to method validation. In HPLC, the LOD and LOQ would often be defined by the signal-to-noise ratio in the baseline, such as in a buffer blank, where the peak of interest would, in the region of the graph where the peak of interest would normally appear. In AUC, the baseline of a blank scan, however, can be artificially smooth because the fitted distribution, often called C of S distribution, is indeed a fitted result. Like the result you see here for a blank by AUC is fitted. It's not a measured signal data versus elution time like it is in chromatography. Therefore, defining LOD, LOQ by this method would result or would lead to values that are not always realistic in terms of the LOD, LOQ. Because the sedimentation profiles, the raw data that you see here, this is the real raw data in an AUC experiment. Because these raw data ultimately define the resolution of multiple species, the resolution that's possible in multiple species, the noise in the raw data should be a strong influence on the LOD, LOQ of species in the distribution profile. In AUC, we have found LOD, LOQ in Charles River by testing a series of standards at low dilution in a manner similar to the slope response type of analysis that one would do in chromatography. An important finding is that we see LOD, LOQs on the order of about 1%, the ability to detect species as low as about 1% abundance, which is consistent with the noise level in the raw scans that you see here, shown above. I may move a little quickly through the rest of these slides in the interest of time, but part of any good validation will also be robustness, the demonstration that the method can remain valid even despite some usually small perturbations in the method runtime parameters. Or if robustness fails, then by doing robustness studies, we want to know within what limits can robustness still be found. We challenged the AUC assay in this regard with respect to these method perturbations, shown here in this slide, and we found the following. With respect to the alignment of sample cells in the rotor, which is a type of robustness challenge, there is very little room for error. We at Charles River have an alignment tool for the purpose of cell placement in the rotor. And when used as directed, data meets precision expectations in accord with what was shown several slides earlier in the precision data. However, if some amount of misalignment is intentionally introduced, we find that even misalignments as small as 1 to 2 degrees will be unacceptable. Sensitivity with respect to this parameter is perhaps not surprising, since it has been reported for protein systems, which I refer to here in a couple of references. But we find here for large species like AAV, which sediment relatively fast, the measurement, the AUC measurement, is especially sensitive to misalignment, and there's very little tolerance. It's an important finding. With respect to the time allowed for temperature equilibration, in other words, the time that the rotor is allowed to sit still or idle in order that the entire rotor body and the samples reach a temperature uniformity throughout prior to beginning centrifugation, we do find the method to be fairly robust for wait times greater than about 60 minutes. However, if equilibration times shorter than this are used, the data can become unstable. Therefore, we do recommend equilibration or waiting times before beginning a run, 1.5 hours or greater for AAV work. The preset centrifugal velocity parameter is generally quite robust. When we dialed in spin speeds that differed by plus or minus about 10% of the nominal value, results still match expected. So we do have a good robustness for centrifugal velocity and any perturbations that might occur in that parameter. Lastly, for robustness, we have the scan frequency parameter, which is still an active area of research in our laboratory, I must say. While we have yet to fully evaluate the effect of different scanning frequencies, or in other words, the density of the data profiles in an AUC experiment that make up the raw data set, we do know, however, we're still evaluating this, but we do know that occasional scans that may be missed, such as an occasional skipped scan that may occur by the instrument, This is a known observation in AUC experiments that there might be an occasional skipped scan in the data set. It appears that one or two skipped scans does not perturb the data set to the degree of affecting the overall conclusions, and the assay is generally robust for this type of occurrence. Now, the presentation thus far has been geared towards showing how the AUC assay can meet many of the criteria of a validated assay, having reasonable results, as I've shown, for precision, linearity, and so forth. Results that can make the case that the AUC assay is indeed fit for purpose for judging the empty-full ratio and other properties. But one overarching characteristic of the assay that must be present these days is the data integrity associated with the measurement and the downstream analysis, etc. Increasingly, the expectation is that the principles of so-called ALCOA here for data integrity are being held to for any assay, but certainly for those assays that are used for lot release or assays on clinical samples. Now, can the electronic records of an AUC assay attain these goals? It's not an easy question. AUC has not had the head start in this area that other assays like HPLC have had. AUC, of course, has primarily been a strictly R&D tool for most of its history. It's been an important part of life science research for decades, of course, but it has never until now been needed so acutely in a regulatory compliant way. With enough depth, however, any gaps, we feel, can indeed be overcome if one has computer systems and IT resources to leverage and to create workarounds where gaps do exist. And I list a few such solutions here that can be put into place. I won't spend a lot of time on them, but they do lend some reliability and compliance to the data handling side of the AUC endeavor. Importantly, having the advantage of large CSV and IT groups, sophisticated groups like that in your company, that can implement layers of security, add-on scripts, etc. to ensure restrictions to the software, adding a layer of security where the software presently may be lacking, including custom scripts, etc., where these software gaps cannot be bridged. Also, where these software gaps cannot be bridged by an electronic solution, we here at Charles River have reverted to paper-based methods, including review checkpoints and robust data form-driven processes for reporting that can show compliance every step of the way. While the paper trail that results can be somewhat cumbersome, especially in an age where nearly everything is electronic, the compliance nevertheless is there, which we've proven. And all this can be done as we have done, although not without a good deal of effort. But reassuring, perhaps, is the notion that in the near future, although not exactly at present, but in the near future, many of the aspects of data integrity may indeed come built in, making GMP-compliant AUC more of an off-the-shelf reality. That's going to conclude, given the time constraints, that's going to conclude what I talk about in terms of the data. There are some additional studies that we can do in it, which I'll just briefly talk about here, in addition to the empty and full characterization that I've focused on, including things like dual wavelength studies at 280 and 260 nanometers. Unfortunately, I don't have the time to present this in any great detail, but you'll have some contacts at the end that you can reach out to us about if you do have any interest. We can do dual wavelength studies, which can help infer the presence of partially packaged capsids, the potential for nucleic acid content in these kind of peaks. And we do some interesting force degradation studies as well at Charles River using AUC, which can be an interesting tool, an interesting complement to other aggregation type assays like SEC and DLS light scattering. So that concludes my part. In conclusion, what I've demonstrated today is that the AUC assay, when applied to the characterization of recombinant AAV drug products, provides excellent resolution of empty, full, and other AAV product variants on par or superior to other methods. It's capable of meeting many of the criteria for a validated method, including precision, linearity, and robustness, and make it fit for purpose for quantifying the heterogeneity in AAV clinical samples. With the proper QA and CSV resources like we have at Charles River, this assay can be made to comply with data integrity standards. And with the right economies of scale, the right labor force, and having enough instruments, you can really run AUC at a throughput stable for most development needs. So thanks, everybody. At this point, I'm going to turn it over to my colleague, Cynthia Swanson, who will talk now in our second part on capsid sorting, TEM, and neural networks. Thank you.
Thank you, Chris. I'd like to give you just a quick overview of a method we've recently developed using artificial intelligence to sort AAV capsids in TEM images. So, as you know, oops, hang on a second. Sorry. Okay. A major challenge in manufacturing AAV vectors is the presence of improperly packaged capsids. Oops. Sorry, technical difficulties here. Okay. So, transmission electron microscopy is a standard method to visualize viral capsids and determine the ratio of full, empty, and intermediate capsids. The manual counting method is slow and labor-intensive, and it can be difficult to consistently identify intermediate capsids. Automated image analysis allows for fast processing of images and a consistent threshold in identifying the categories. This project addressed whether or not we could successfully automate AAV capsid sorting. Traditional image analysis uses parameters such as color, size, and shape to distinguish objects of interest. As you can imagine, monochromatic TEM images give us fewer tools to use in analysis. The addition of AI platforms gives us another option. We recently acquired both the Aphoria and Visiopharm AI platforms, allowing us to utilize neural networks for image analysis without having to become computer scientists. Our hope is that in applying neural networks to these monochromatic images, we'd be able to accurately sort AAV capsids. Just a little background. Deep learning is a subset of machine learning, which is a subset of artificial intelligence. Artificial intelligence may be thought of as a computer system's ability to perform tasks that normally require human intelligence, basically a program that can sense, reason, act, and adapt. In machine learning, algorithms improve as they're exposed to more data without being directly programmed. Deep learning utilizes multi-layered neural networks and vast amounts of data. Both the Aphoria and Visiopharm platforms use convolutional neural networks. These layers of nodes are loosely patterned after neurons in the human brain. We're using a form of supervised machine learning. An expert will annotate the images for training, and the deep learning networks perform automatic feature extraction. A key component of getting a good result is in starting out with very good ground truth. I like this example. We all recognize handwritten numbers, even when there's variation in the handwriting. Notice that all the zeros look very similar, as do the ones. But once you get down to the 5s and 6s, and definitely the 8s, there's a lot more variation. What we can easily recognize as an 8, the machine may not be able to distinguish without a lot of repetition and a lot of variation being presented during the training. In order to train the neural network, we need high-quality TEM images of the capsids. The preparation of a sample is extremely important in regards to the density of viral capsids, fixation, and negative stain. And here's one of our TEM experts collecting images. Sample preparation is key. We've collected a variety of images to use in training the neural network. And here's the result of training. The green arrow shows full capsids, which exclude the stain. The red arrow indicates an empty capsid containing dark negative staining, and the blue arrow shows an intermediate capsid, which has a bit of stain. Visiopharm is a validated platform for image analysis. As the TEM images are also validated, we're able to offer a GLP-compliant analysis. Visiopharm DeepLab has worked very well in identifying and classifying the capsids. Even with assorted artifacts, this app has worked very well. So, in conclusion, TEM with negative staining allows us to visualize the quality of the capsids as well as the quantification of the full, intermediate, and empty ratio. CENs are able to quantify difficult to distinguish objects in images previously not suitable for traditional image analysis. And sorting of full, intermediate, and empty AAV capsids is fast and accurate using neural networks. So thank you for your attention, and we're ready to answer any questions you may have.
Okay, great. So the first question is, what kind of sample amounts are required for R&D and GMP testing by AUC?
Yeah, thanks, Leah. That's a good question, and it's a question we get a lot. Practically speaking, for routine R&D tests, we only require several hundred microliters. If the titer is about 1 to the 13th, which is becoming a common titer level to be able to provide, about 100 to 200 microliters is sufficient for a routine test in duplicate at the R&D level. Now, to do a full GMP validation, that's a lot of work. That's many tests, precision. There's a lot of repetition there. So we're using, as large as it may sound, several milliliters of the sample if the titer is 1 to the 13th. Fortunately, clients do seem to be able to provide at these higher levels now as manufacturing processes seem to be improving and streamlined, and production in bulk is more and more possible. Thanks again for that question. Yeah.
Do you have some data based on LVV? Lentiviral vectors, I'm assuming.
Lentiviral vectors. Do we have any data on lentiviral vectors? We have studied other viral vectors other than AAV. Adenovirus, we've studied that. Some HSV. I believe we have some data on lentivirus. Not a lot, but AUC as a platform is certainly amenable to any kind of sedimenting species, and even these very large viruses sediment quite fine. One can tune the centrifugal velocity to the size of the species, so it's certainly quite within the bounds of what AUC can do.
And what wavelength do you use to measure antibodies?
For antibody detection, that's a pure protein, and it's usually available in sufficient amounts where we'll study at 280 nanometers. That's a common absorbance wavelength for an antibody in an AUC experiment.
Go ahead.
Incidentally, we use a lower wavelength that you might have noticed from the slides. In AUC, we use a lower wavelength of detection in AAV particles because of the stronger absorbance there, and the relative equivalency between empty and full capsids at that lower UV wavelength. Go ahead. Sorry.
Okay. And what is the largest sample size in AUC that AUC can process?
The largest sedimenting species? That's a good question. One can make some inferences into what would be the largest size thing that you can see. In some of the graphs that I've shown on the AUC size distribution profile, you'll see that an empty capsid at about 60S, the full capsids near 100S, and those are megadalton size. An AAV capsid is about 3 megadaltons when empty, maybe about on the order of 5 megadaltons when full. And so we use this terminology S, the Svedberg's, the sedimentation coefficient units, which relate to mass. And so if 100S is about 5 megadaltons, you can see in that plot that I've shown that we're fitting all the way up to 100S, 150S, 200S, which is tens of megadaltons. So certainly you can see things in the tens of megadaltons, which covers many viral species. things on the order of whole cells might start to become too large because there's a limit on how slow you can spin these centrifuges, and there's also a limit in terms of when you can first start detecting, and this is important, when you can first start scanning for data, because it takes a discrete amount of time to go from the rotor being at full rest at 0 velocity up to your target velocity. That happens gradually; it doesn't happen instantaneously. And as the rotor is approaching that target speed, it's very difficult to collect data in that interim period. But sedimentation is still happening, so very large things will sediment during the acceleration phase and can be lost. So again, those limits are probably ruling out whole cells, but many viral species are quite amenable for ASSAY.
Okay, so we have time for just one more question. There were a number of questions that we were not able to get to. Also, if you go ahead and type in any questions now, all those questions will be passed on to Christopher and Cindy, and they will follow up with you directly. So the last question is, are full and empty AAV stocks or standards available for customer use?
Are full or empty stocks available for customer use? We do not produce at present off the shelf any kind of standards. We feel that some commercial manufacturers may soon start to have this in an analogous way to how for monoclonal antibodies there are some now mass-produced IgG standards, some NIST control standard antibodies. We may soon see the day where that's readily available for AAV. It's not at present. We are very interested in that as well, and are looking to be have a key role in it. More on that to come, I suppose. But unfortunately, at present, no.
Okay, great. Thanks, Chris and Cindy.
Thanks, Leah. Pleasure.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.